some reason my upload to YouTube is so slow. It takes like 4 days. So I will shoot it faster. Okay. Lymphatic system. So lymphatic system is our immunity. And first we are going to look at pathogens. Actually, the word pathogen and antigen are quite similar because both antigen and pathogen mean something foreign to the body. So antigen is something foreign to the body. So pathogens kind of scarier than antigens. Antigens also can be scary because it causes some pathology. And we're going to look at 3 groups of pathogens: bacteria, viruses, and prions. Things that infect our body. So, first of all, what are bacteria? Bacteria are cells, individual cells. You know, when they get into our body, they absolutely love it, and they have no idea how damaging they are for us. They live, bacteria do not have nucleus, and their DNA is somewhere swimming in cytoplasm. And when they divide and grow, they will release yucky chemicals into our body, and we don't like those chemicals. And, bacteria will disrupt our metabolic processes. What is the #1 drug to treat bacteria?
Penicillin.
Yes, penicillin, and antibiotics, amoxicillin. For bacteria, for any cell, in order to develop, it needs to build its own proteins. Proteins are built where? On ribosomes. So how of those antibiotics kill ribosomes? How you can kill ribosome? Antibiotic, which is a chemical, goes there, gets stuck in there, and ribosome will stop working. So bacteria cannot make their own proteins, they cannot divide, they will die eventually. The second half of antibiotics goes to DNA and stops transcription. Once again, it's related to gene expression. Gene expression includes 2 steps: transcription and translation. DNA and RNA. This one is transcription. And then protein. This one is translation. So it kills either here or here, disrupts the process of gene expression, bacteria will die. Now, what are viruses? Viruses, well, they come in different shapes, but I like the picture of virus that looks like a spaceship. Somewhere in the book, and it's 6 legs. And here's DNA. There's a variety of viruses, some carry RNA, so they build DNA and so on, but it's more complicated processes. We are not going to talk about them. We take one of the simplest viruses, and the virus is made of protein capsule, course, DNA, genetic material, because nothing can develop without genetic material. And it lands on our cells, on our tissues. This is us. This is host cell. And it injects its DNA into our cell. So in comparison with the bacteria, viruses do not have their own organelles. Viruses are considered half dead, half alive. Bacteria definitely, they are alive. They grow, they develop, they divide. Viruses are half dead. This is like strange definition from the monster movies. But they can be there for like years and they'll come alive when they meet us. So when virus injects DNA into our cells, it uses our machinery, our ribosomes, to synthesize capsids, and to synthesize DNA, our enzymes. And that's why we cannot kill virus with antibiotics. What are we going to kill? Our own enzymes? We will die as well. So we need a special system that is going to fight viruses. First look for them inside of our host cells, and then somehow deal with the problem. And this system is lymphatic system. We're going to talk about immunity, vaccinations, and lines of defense. So prions, the third class of pathogens, prions, proteinaceans, protein, PR is from protein, infectious particles. These are misfolded proteins. Normally, when protein loses its shape, it loses its function as well. But there are some strange cases when the protein, in some cases, when the protein changes its shape. It can be folded this way or this way, and under certain conditions, all of a sudden it got folded the wrong way, and all of a sudden it got some monstrous activity, yucky activity, mad cow disease, and it will start misfolding the other proteins. So these are the strangest thing, and it has a relation to three-dimensional shape of the proteins. So, okay. Now, yeah, everything misfolded also must be identified and removed from the body. Now, how our body can protect us? What are the lines of defense? First line of defense is our skin and liquids that it releases. Our saliva, you know, there's also some areas that are not skin but still epithelial cells with the ducts, and our body fluids, they have slight protective effect. Like, you know, the dogs always lick their wounds, not just to clean it, but because saliva has a little bit of lysozyme, it has a little bit of basic pH, bacteria do not like it. So it's physical barrier and chemical barrier. So the first line of defense is skin. physical and chemical barriers. Second line of defense is non-specific defense. And we are going to talk about regular cuts. We are going to how they heal, and we are going to talk about blood and what is in there. Okay. So okay, let's finish this concept. Put here non-specific defense. Sorry, I have to find the little corner to write. Defense, and put here a word neutralization. This is the name of the blood cell that non-specifically kills everything foreign. And the third line of defense, we're putting here specific defense, and we're putting here, just write it for now, B and T cells. And then we write the word lymphocytes. Lymphocytes. Because B and T cells are the cells of the lymphatic system, and they are called lymphocytes. Let's talk about second line of defense. First of all, how cardiovascular system is related to lymphatic system. There are blood vessels and lymph vessels. Blood vessels can carry red blood cells and white blood cells. Lymph is a special liquid, and the lymphatic vessels, they are positioned very close to cardiovascular system. See blood capillary. Let's look at that. And here's a lymphatic capillary. Lymphatic system is in our body to transport fats, so not too many fats get into blood, and to be special pathways for white blood cells, but red blood cells never get into lymphatic system. Lymphatic system also delivers tissue fluid, so all of our cells will be washed in the special tissue fluid. But the primary purpose of lymphatic system is to produce, making, and house white blood cells. White blood cells, they can squeeze into every space in the body. They're just those guys. Red blood, see? This is a blood capillary. Page 1. Yes. You have to tell me such things. When I see here and you don't see it over there. Look how white blood cells checks everything around in the tissues, and then it squeezes into blood capillary. This is epithelial cells, squashed squamous epithelium, and travels with the blood. Okay. Blood composition. I want you to know function of red blood cells. Once again, it's just a summary of 2 chapters, and we're kind of going quickly through the material. So I'm pointing out the most important things. Red blood cells carry oxygen. I want you to write oxygen like O2, because there are always 2 atoms in the oxygen molecule. White blood cells fight infection. Now 70% of the total number of white blood cells are neutrophils. Neutralizers. Neutrophils. Did I write the word neutrophil correctly? You will have it in the vocabulary. Yes, neutrophil. Right here is white blood cell, non-specific. And 30% of white blood cells are called lymphocytes. 2 types of lymphocytes, B and T cells. So a lymphocyte, once again, it's in your vocabulary, is a white blood cell that provides specific defense. Why our blood is red? Because of the red blood cells. What exactly is there in red blood cells that makes it red? Oxygen, not really. There's something that is related to catching oxygen that is red by its nature. Hemoglobin. Okay, hemoglobin. In order for hemoglobin to catch oxygen, it needs what? If you don't have a specific element in your diet, you get anemia. This element is iron. Okay, what we're going to write here is hemoglobin plus iron necessary for carrying oxygen. Now let me show you a picture of hemoglobin. Okay, here's the iron. Here's the blocks of iron sitting there. And the whole hemoglobin needs to catch iron, and iron catches oxygen. Now, what is sickle cell anemia? Is anybody familiar with that disease? Red blood cells live for 120 days. Sickle cells anemia, the red blood cells look like sickles. Why? Because of the misfolded hemoglobin. See how nice and compact it is here. So I'm going to draw it 1, 2, 3, 4. That is your hemoglobin. Here's another hemoglobin sitting nearby. Everything nice and compact. In this case, in the case of sickle cell anemia, I'm going to draw hemoglobin a little bit misfolded. Not that misfolded as in prions or something that causes infection, just misfolded. Does it catch oxygen? Yes, it does. It's able to have iron, and it can catch and transport oxygen. But the problem is that this shell, this shape of gets stuck in blood capillaries, and the cells will get damaged, and they live only 90 days. So, and it's very painful when all of a sudden your blood cells will get destroyed. First, you don't have enough oxygen. Second, it's just painful. And third, your immune system, your red bone marrow, which is a place that produces all kinds of blood cells, it works overtime to create enough of those sickle cells. But in evolution, it happened to be helpful. Did you hear about the disease malaria? Okay, and this is a parasite that gets into red blood cells. And this parasite will destroy red blood cells when it's ready to come out, so a person will have a fever. But if the parasite gets in sickles, these cells, they will die in 90 days. And parasite has a cycle of 110 days. So the parasite cannot reproduce in these cells. And that's why sickle cell anemia, you can see it mostly in the regions that are affected by malaria, because this disease will help people survive. So normally, it's very hard to live with that. But when the decision is to live or die, that lives. Normal dies in the areas with those mosquitoes and malaria stuff. Okay, hemoglobin, iron. Hemoglobin is a protein, must be folded correctly. Now, blood is, okay, which type of tissue is it? Bravo! Connective. Fluid connective tissue. And the basic structure of any connective tissue is cells and matrix. And this matrix, okay, it can be red blood cells, white blood cells, and this are water and protein fibers. So remember that blood is not only red blood cells, white blood cells, but also protein fibers. Hemophilia. Hemophilia is a disease related to the lack of certain proteins in the blood, because those proteins are used in clotting your blood. There are like 21, 23 factors that are participating in blood clotting. One is missing, the blood doesn't clot. Hemophilia. Remember that there are proteins there, also some salts, the nutrients. Okay, who's going to carry your nutrients from your stomach to your brain? Of course, it's blood. Of course, it will transport amino acids, glucose, everything is there. So there are chemicals. And if there are a lot of chemicals, there are a system of buffers. Buffers are pH shock absorber. So something simple chemicals that keep pH in a neutral level. That is blood composition, which I covered in more detail in right here. Let's go over vocabulary that you need for them. All blood cells are created in red bone marrow. Where is it and what is it? Red bone marrow. Did you ever cook soup from the bones? Okay. So if you take a bones, okay, like let's say, and cut it. Of course, here's a cartilage. So here's the tough part, but some part at the end of the bones will look like a sponge. And in this spongy part, there is a very, very important tissue, conglomerate of cells that is called red bone marrow. That's where all blood cells are born. Because when you need, when your immune system is malfunctioning, you need a transplant. And red bone marrow is one of the most popular transplants. So red bone marrow, how do we define it? Birthplace of all blood cells. That's what we need to know for now. Located deep in the bones. Okay, next one. Platelet, thrombocytes. Oh, I apologize, I forgot to add them. Here, thrombocytes, thrombocytes. Cell pieces used in clotting, blood clotting. What does it mean, cell pieces? They are created from a special ancestor that once again is born in red bone marrow. And then this precursor will split into millions, into thousands of smaller particles. Like, you know, you take a big plate, that's basically what it is. One big cell in the beginning, done. Because they are used in clotting. They are not like trash or something. They are specific part of blood composition. Please look at the picture. Okay, 115. And here, platelets. You know, some people go to the hospital to get an infusion of platelets and investigate the lymphatic system that don't matter malfunctions. So platelets are thrombocytes. The word thrombos, like block, clot something. Next one, neutrophil. Okay, let's skip neutrophil for now. Antigen, foreign substance to the body, to our body. Clotting factor, protein in the blood. Fibrin, another protein in the blood. Fibrin is also used in clotting. The difference between fibrin and clotting factor is kind of minimal from our point of view. Fibrin is one of the enzymes that initiate response to some wounds and initiate the process of clotting. If I had more time, I would have talked more about fibrin. Now we have antibody, which we are going to cover in lymphatic system because there is also antigen and antibody thing. What we have left from this vocabulary is neutrophil and lymphocyte. And they're both right here. These are both white blood cells used in defense. So let's look at non-specific defense. Oh, by the way, while I was going through the book, I found this picture, and let's look how the process of clotting looks like. Page 121, blood vessel is punctured. First, it's platelets. So it's the smaller pieces of one huge cell, like sand, get sucked in and get stuck right here. But they don't have anything to hold on to, so to make this plug more secure, there's certain metabolic process taking place right there. And you see the word fibrin right here. And fibrin is just those long protein fibers that assists in blood clotting process, and then the other clotting factors will aggregate right there. Okay. Finally, moving right here. You can see bacteria. This is the bacteria in comparison with our cell. This is a virus, different kinds that I draw on the board, but works the same way. Here's our lymphatic system. Okay. Lymphatic system. Primary organs. Red bone marrow. Deep in the bones. Deep in bones. And thymus, which is located right here. Thymus sits right on top of the heart. If we talk about life cycle of white blood cells, they need a place to be born, just like we are. And this is red bone marrow. And then, like in every place, let's say Bangor, there are people that go to college, and there are people that do not go to college. Or let's say university. And thymus is a special university for part of the cells that decide to be educated. So the easiest way to remember why those lymphocytes are called B and T. B cells born in red bone marrow and never go anywhere. T cells born in red bone marrow and went to Thymus University. They are graduates of Thymus University. Now, what is the program of study of Thymus University? Cells there learn to distinguish the cells of our body from the cells that come from outside. Or let's say somebody gets a transplant, and that transplant is not correctly matched. Who is going to say it's not our cells? That's something foreign in our body, let's get rid of them. They're the T cells. Graduation. What is the graduation rate? Very, very low. And if you don't pass the exam, die. That's the bad thing about Thymus University. But sometimes, you know, there are not good students can graduate, and then you have allergies. Because allergies is when your lymphatic system reacts to something they shouldn't be reacted to. Or when, let's say, arthritis. It's the cells of your own body, but those yucky graduates of Thymus University think, oh no, we don't look like ours. So, thymus. That's where the cells learn to distinguish first our cells and not our cells, but those that look like our cells, like from the other human. And the second program of study is to distinguish our cells that are healthy and those cells that are infected with virus. Because when virus infects our cells and start using our machinery to produce its own, capsules and DNA, our cells will slightly change. And those T cells, they must detect those changes cells that house the virus. Because not ours anymore. We need to kill our own cells of our own body so virus will not get a time to develop and infect more of our cells. And they will kill the cells of our own body, but only those that are infected with the virus. Bacteria is outside of the cell, much easier, detectable by B cells. T cells will recognize the enemy inside of our cells. The enemy thinks that it's kind of hidden, but no, those cells must bind. All right. Lymphatic. Now let's talk about non-specific defense or inflammatory response. So we put here the neutrophils, 3 lines of defense. Put here inflammatory. Response. Inflammation. You always get the same inflammation even if you get different bacteria. And then if the inflammation is not enough, then the third line of defense, a lymphatic system, the cells will get activated. But let's first see what happens in inflammation. You got a cut. See the cut? Right here. Immediately, first of all, what happens when you get a really, really bad cut? The blood vessels dilate because of histamine. Same process could be activated in allergies, so you take antihistamine drugs to shrink a little bit your dilated blood vessels. But remember that if there is a cut or infection, the blood vessels will dilate. To bring more blood to the place of the injury. Why? First of all, blood will bring heat. Everybody imagine how you get a cut and how hot is whatever place of wound right now. Remember, blood brings heat. Heat will kill some of the bacteria. Bacteria do not like heat. 42 Celsius grade, which is probably 1 or 6, 7, most of them die. It's a heat shock for bacteria. Second, of course you get a lot of pain, because if the blood vessel dilates, it will start pushing on the nerve endings. You get a lot of pain. You know that you need to do something about it. Second thing, a lot of white blood cells come with the blood. And these are first responders. Find the word neutrophil in somewhere in your notes and put first responders. So they are coming. See how this neutrophil will grow a mouse, and by endocytosis will gobble up bacteria. Then, of course, when your wound is healing, and if there was an infection, you will get a lot of pus. Have respect to this liquid. These are bodies of dead neutrophils. Okay. They died, gobbling up your bacteria. And if neutrophils feel that there are not enough of those, or they keep eating bacteria and nothing still happens, or it may be a virus that they start screaming by calling for the help of the third line of defense. Please consider it as a war. And this message is called cytokines. This is a message. Help me, SOS. Help me, lymphatic system. There is an enemy in our body now. So remember that neutrophils are first responders. White blood cells are mostly neutrophils, 70%. 30% are lymphocytes, and a bunch of others, they just a smaller percentage of white blood cells. There are different kinds. Some are specialized for worms or something else. But anyway, if you look at that picture, that is a lymphocyte, about 30%, and neutrophil, 70%. These are red blood cells, which totally are not concerned about infection. They carry oxygen, and good luck with that. But there are some others that take care of more specialized parasites, a small percentage. Okay, now we have 15 minutes to talk about vaccinations and B and T cells. Vocabulary. White blood cells are mostly neutrophils and lymphocytes. Red bone marrow produces blood cells, and thymus, let's put the word educates blood cells. B cells and T cells. B cells just born in bone marrow, because B is for bone marrow. T cells, T stands for thymus. They are lymphocytes. Okay, and B cells and T cells belong to specific defenses. As you can see, the second page just gives you the most important summary of the chapter. We're going to talk about mechanism of activation, of immunity activation, and how we get an immunity. And I need a specific picture from the book. I'm trying just to explain it a little bit simpler. Okay. Mechanism of activation. We're going to draw a diagram. I'm drawing a variety. Let's start with B cells. I'm drawing a variety of B cells. And remember that a cell membrane have a specific so-called receptors, specific proteins. And I'm drawing here a different shape of those proteins to show that our red bone marrow creates a variety of B cells which are very similar to each other, except that they have a different receptors, proteins, on their cell membrane. So now I'm going to draw antigen. Which is a foreign object. Foreign object may mean part of bacteria, virus, which haven't got into cell yet, or some toxic substance. So everything that comes into body needs to be checked. Okay, and of course every foreign substance is being checked, and in most cases it fits one of those receptors. So do you see a cell where this antigen can potentially fit? Okay, let's say it goes and fits like right here. And I'm writing the word fits. And the next, what happens next? The cell is selected by antigen and activated. It's called clonal selection because this cell actively divides. What does it mean to be activated? It means division. And this cell produces two types of cells. They're called plasma cells and memory cells. Now the difference between plasma cells and memory cells are identical to original cells, but we just have a little bit more of those. They're going to remember this invasion, this infection. Plasma cells are huge and they immediately will start producing the receptor that is no longer sits on the cell membrane, but goes into the blood. As a result, what will happen? Here's the antigen. And in this case, it's called antibody. There are viruses infecting the body or infectious particles that those, in other words, antigens that will be found by those antibodies and marked for destruction or obliterated so much that they will be eliminated from the body very, very easily. And the last step, when finally all antigen are taken care of, and we have a lot of plasma cells. You know, you're sick. You are very, very sick, and your body is working on creating those plasma cells and creating this specific protein, which is an antibody, in fighting virus. And then you finally fought it, and you won, and you don't need plasma cells. Yeah, that's life. So they self-destruct. Apoptosis. Self-program or programmed self-death or programmed self programmed death. Okay, once again, activation of the cell happens when antigen binds to receptor. So what do we have for? Here, clonal selection in vocabulary. Antigen binds to the receptor. Apoptosis programmed self-death or programmed self-destruction. Receptor protein on cell membrane. Testing antigens. Antigen is foreign substance. Antibody is, here's antibody. Antibody, a protein similar to receptor that binds antigen. Let's put here binds antigen, marks it for destruction. Marks it for destruction. Now, vaccinations. Now you can put vaccinations into this diagram. What is vaccine? Vaccine is dead or non-functional virus, or part of the bacteria, or something that, if it was viable and healthy, can cause an infection. But you get a vaccine and you may even get like inflammation. It cannot cause a disease, but it can activate your immune system exactly the same way as disease activates your immune system. So it will create plasma cells and memory cells. Plasma cells will self-destruct. Memory cells will remember the invasion. It's no longer just one cell out there monitoring. There are a bunch of memory cells. So second time, can virus come into your body? Yes, it can. But it will be detected so quickly by memory cells that it cannot run the course of this disease. Because this process will take a week. That's why flu, you're down with the flu for a week. Okay. Now, there is a difference. This is for antigen. This is antigen that is outside of host cell. And B cells will take care of such antigens. Now, what should you do if antigen is in host cell? We have T cells. So, in your vocabulary, this concept is reflected in a sentence. B cells are responsible for antibody-mediated immunity, and T cells are responsible for T-cell mediated immunity, or antibody-mediated immunity, and T cells are responsible for cell-mediated immunity. B cells will produce antibodies, T cells will not produce antibodies. But what I'm drawing here is a virus inside of the cell. And cell fights, this is us, this is a host. And then the cell will try to fight it. It may be successful, it may be not, but it will change slightly, and it will try to remove somehow parts of the virus, parts of the pathogen. And it will have a certain change in the membrane. So now, what you need to remember that T cells are blind. After graduation from Thomas University, they are blind. They go and they kind of, check everything around, and they need a helper. They always work with a helper, like in past time, every blind man needs some, boys that, go after someone with a hand on a shoulder, and page 144. So on page 144 you see a picture that shows different T cells. Then it shows a helper, well, not a boy, but some helper that digested this bacteria and exposed to T cells some certain changes on the membrane that host cells will get. And then clonal expansion reflects the activation of T cell. T cell becomes really, really angry, cytotoxic T cell, and it has its own weapons, gramzymes and perforins, very scary names, which are proteins that will be used to destroy host cells that house the virus. Okay, the process. So let's. You're gonna be the one that, like, already digested the bacteria, that macrophage, and you're gonna expose a part of bacteria for me, and I'm just a blind T cell that goes and checks everybody. Okay, let's say some cells will have a virus. That's a virus. That's a virus. So, and you are a good one, and you just helpless ones that cannot find it, or they can like slightly fight it. So, and I'm like this monstrous T cells with gramzymes and. Okay, and first I'm going, I'm checking. Okay, this is normal. Okay, and here's my helper that comes to me and gives that to me and says, here. Okay, got it. So I got my weapons, perforins. Go and check. Do you have that? Do you have that? Oh, there's virus there. Done. Normal. Oh, virus there. My gramzymes, perforins. Done. All right, virus died inside. Okay, and that's what all immunity about. Activating a certain kinds of B and T cells that already know what the antigen is. Vaccinations are about giving you dead or non-functioning virus. The cells should have an extra chance to go through the process and I have to let you go, unfortunately. I will talk with you for hours. Oh, let's finish the last sentence of vocabulary. 3 ways to get immunity are: get sick, suffer, but then you get working immunity. Second, get vaccine. Don't suffer, but you get the same stuff. And third way to get immunity, when you get just the antibody. How do you get the antibody? By shot. Because, for example, you go to some area with a dengue fever or whatever, and you don't have 2 weeks to wait when you get those cells. So you get just the proteins. But if you get the memory cells, you will remember the infection. You maybe need a booster shot or something. But passive immunity, when you just get proteins, will be done in 3 months. Because proteins will be destroyed and taken out of the body. But memory cells can create a lot of proteins. Okay, and if you're getting proteins, it's called passive immunity. If you have memory cells, it's active immunity. So write me emails. Good luck preparing for an exam. Interferon. Interferon is a drug. It's actually when your host cells that got infected by virus, that's what you put out as a message to the other cells. Get prepared, the enemy is coming, virus is coming. So an interferon is a protein. It could be made by biotechnology. That's why it's a drug, and if your liver is affected by some kind of virus, they will give you interferon to make those healthy cells maybe a little bit more healthier, to prevent the virus from